Welcome back for another deep dive. This time, we're tackling a topic that's seriously fascinating. We're diving into the world of gene expression.
And trust me, it's way more complex than you probably think.
Yeah. We'll be drawing insights from a really cool study called mRNAs, Proteins, and the Emerging Principles of Gene Expression Control.
It's a great deep dive into how genes actually make you.
Ready to unlock some knowledge?
Absolutely. Let's do this.
Okay, so the paper starts off pretty basic. Gene expression. It's how your genes create your traits, often by making proteins.
Right, the classic definition. But it's kind of misleading. It makes you think it's just a simple process, like more mRNA, more protein, end of story. But it's not that simple at all.
So there's more to it than that, huh? What makes it more complicated?
Think of mRNA like a blueprint, right? And proteins, those are the actual building.
I see where you're going with this.
You need both. Having tons of blueprints doesn't mean you've got a skyscraper going up. You need the materials, the workers, the whole shebang.
I like that. So if it's not as straightforward as just like cranking up the mRNA volume knob, how do scientists actually study this relationship?
Well, they look at what's called correlation, basically how well the amounts of mRNA match up with the amounts of protein.
Okay, so how strong is this correlation? Can we actually predict protein levels just by looking at mRNA?
There are actually two types of correlations scientists look at. Across-gene correlation is like comparing blueprints for different buildings at the same time. So you see if the amounts of each blueprint match the number of actual buildings you have.
So are we talking about like one blueprint per building? Or can you have multiple blueprints for the same structure?
Exactly. It gets complex. And then there's within-gene correlation. This one's trickier. It's like following the construction of a single building over time. Does the amount of blueprint predict how much of that specific building gets built each day?
Interesting. So is the amount of blueprint like a good indicator of building progress?
You'd think so, but that's where things get surprising. The correlations are often weaker than we expect, especially within gene. There's clearly a lot more to the story than a simple one-to-one relationship.
Now I'm really curious. What is going on then? How does it all actually work?
Well, let's break down the gene expression pathway. It's a journey, not a sprint. We start with DNA.
The good old double helix. The instruction manual for life.
Exactly. Then we move to mRNA. That's the messenger molecule. And finally, we arrive at the protein. Each step influences the next. What's crazy is that a tiny amount of DNA can ultimately lead to millions of protein molecules.
Wow, that's like a single acorn sprouting a whole forest.
Right. And with that kind of amplification, there are all sorts of points along the way where the process can be controlled.
So it's not just an on-off switch for each gene.
Nope. It's more like a volume knob with a ton of different settings. Transcription, where you make mRNA from DNA, is super important, especially for big decisions like what kind of cell to become.
Like, should I be a skin cell, a brain cell, a muscle cell?
Exactly. But then there's also this whole other level of control. Post-transcriptional regulation, that's where things get really interesting.
Okay, I'm listening. What does that involve?
It includes controlling translation. So that's deciding how much protein is actually made from that mRNA. And it also involves protein turnover, how long those proteins stick around.
So it's not just about making the proteins, but also how long they hang out in the cell. Makes sense. Things change. You got to be adaptable, right?
Right. And the cell needs to be able to respond quickly to those changes. The study actually gives a great example. When cells are stimulated with LPS, that's a molecule found in bacteria.
So the cell is facing a threat. It needs to respond fast.
Yeah. What's interesting is that even the levels of housekeeping proteins, the ones essential for basic cell function, even those change.
Oh, so even those core proteins are affected by this bacterial molecule. How does that work?
Well, instead of making more mRNA through transcription, the cell mostly tweaks translation and protein degradation rates. It's like changing the amount of time you cook something or the temperature of your oven. You can get two very different cakes from the same recipe.
Whoa, mind blown. So it's all about those subtle adjustments after the mRNA is made.
It's a super efficient system. We used to think transcription was the main control point, but it turns out that post-transcriptional regulation is playing a much bigger role than we ever imagined.
It's like the cell has all these backup plans. So cool. But are all these steps in gene expression equally fast? What if you need a protein in a hurry, like to fight off an infection?
Great question. Cells are incredibly smart. They've figured out ways to optimize speed. Now, transcription and translation do have limits. You can't just churn out proteins infinitely fast. There are physical constraints.
Sure, there's got to be some kind of speed limit, right?
Right. But cells can have certain mRNAs ready to go. Like imagine you've prepped all your ingredients for a recipe. When you need that dish, boom, you can whip it up fast.
So it's like meal prepping, but for your cells.
Exactly. And then there's the other side of the coin. Getting rid of proteins, sometimes that's the speed bump. Some proteins are super stable. They hang around for a long time.
Like those leftovers that just won't quit.
Exactly. But cells have a solution for that too. Some proteins, especially those involved in dynamic processes, have really short lifespans.
It's all about balance then. Speed things up when needed, slow things down when necessary. This is amazing. This whole system, this dance of gene expression, it's what allows our bodies to adapt to literally everything, right?
Yeah.
Infections, changes in our diet.
You got it. It's all about flexibility and control. And that brings us to another really fascinating aspect of gene expression. That protein to mRNA ratio we talked about earlier.
Right, so we said it gives you a rough estimate of protein abundance across different genes.
Yeah, but it's not reliable for predicting protein changes within the same gene. Under different conditions, it's like something is intentionally smoothing things out.
Like a buffer or something.
You got it. And it's called, you guessed it, protein level buffering. The cell has these awesome ways of ensuring things run smoothly, even when there are bumps in the road. Think of it as a shock absorber for your cells, keeping those protein levels stable even if mRNA levels go up and down.
Protein level buffering. It's like the cell has its own internal quality control department. So even though mRNA levels might fluctuate, the cell prioritizes keeping the protein stable and functional. Because at the end of the day, those proteins are the ones doing the real work, right?
Exactly. It's not just a theory either. There's real evidence for this. Remember those omics technologies we mentioned?
Oh yeah, those super powerful tools. They're like giving scientists X-ray vision into the inner workings of our cells.
Using these techniques, scientists have discovered some pretty amazing things. For example, in yeast cells, even if you delete one copy of a gene, the protein levels often stay surprisingly stable.
Wait, so even with less DNA to work with, the cell still manages to churn out the same amount of protein. How is that even possible?
That's the beauty of buffering. In that particular case, the cell compensates for the missing gene copy by increasing the production of mRNA from the remaining copy. It's like, okay, we're down a worker, so everyone else has got to pick up the slack.
It's like this amazing backup system.
Yeah.
It ensures that everything keeps running smoothly, even when things go a bit haywire. It's so cool.
Right. It highlights how intricate and adaptable gene expression really is. It's not rigid. It's constantly adjusting and compensating.
So how does this buffering actually work? What's the magic behind the curtain?
There are a couple of key mechanisms at play. One is called autoregulation. Basically, some proteins can control their own production or degradation.
Like a thermostat, but for proteins. They can sense when there's too much or too little, and they adjust their own levels.
Precisely. And then there's another mechanism. It involves something called orphan subunits. Imagine proteins as complex machines, right, with multiple parts. If there are extra parts floating around that haven't been assembled into a complete machine, the cell gets rid of them. It's all about efficiency.
It's like a well-oiled machine. Everything has its place, and everything is working together. So this buffering system helps to keep things running smoothly. It makes sure we have the right balance of proteins and that everything functions as it should.
Exactly. And this brings it back to you. This buffering system is working inside your body right now, helping you stay healthy and functional despite all the genetic and environmental variations you encounter every single day.
It's pretty mind-blowing. But now I'm curious, if we zoom out a little bit, in the grand scheme of things, who wins the information prize? Is it mRNA or protein? Which one gives us the most valuable insights?
That's the big question, isn't it? But it's important to remember that both mRNA and protein measurements offer really important and unique insights. It's not about picking a winner.
Okay, so no clear champion.
Think of it this way. mRNA measurements are like peeking at the blueprint, right?
Yeah.
They tell us which genes are being transcribed, how much, and when.
So mRNA is like seeing the architect's plan before the building goes up.
Exactly. But then protein measurements, those are like seeing the finished building, the functional structure. They're closer to the action, to the function of the genes.
So it's like appreciating both the process and the end result.
Exactly. Combining both types of data, that's where the real magic happens. It's like having the blueprint and the finished building.
Yeah.
That gives us this much more complete picture of how genes are expressed and how that translates into real-world function.
It's like having all the pieces of the puzzle. We need both mRNA and protein data to understand the full story of gene expression. And this understanding is crucial for so many things, right? Like understanding development, disease, even aging.
Exactly. And speaking of understanding, we've only just scratched the surface of this topic. Let's dive into what this new knowledge about gene expression means for the future of healthcare.
Okay, yeah. This is where things get really interesting. Let's do it.
So this whole intricate dance of gene expression with all its checks and balances, it plays a crucial role in everything, from how we develop to how we fight off disease.
It's pretty wild when you think about it. All this happening inside of us every second of every day.
Right. And that brings us to something really exciting. Personalized medicine.
Yes. We touched on this a bit earlier, and I'm dying to know more. So, if we can understand how our genes are expressed, could we actually tailor treatments to each person's needs?
That's the holy grail, right? And the field is moving towards that goal really fast. Imagine a world where treatments are customized to your specific genetic makeup and how your genes are working.
It sounds futuristic, almost like science fiction.
Yeah.
But is it actually achievable?
It's closer than you might think. Our understanding of gene expression is growing exponentially, and that's opening up all sorts of possibilities for personalized medicine.
Okay, I'm officially intrigued. Paint me a picture. How could this knowledge actually be used in a doctor's office or a hospital?
Let's take cancer treatment. Right now, many therapies target rapidly dividing cells, but that includes healthy cells too, not just the cancerous ones, hence the nasty side effects.
Right, it's like that old saying, you can't make an omelet without breaking a few eggs. But in this case, we're talking about pretty important eggs.
Exactly. But imagine if we could actually pinpoint the specific genes and proteins driving the growth of a particular tumor. Then we could develop therapies that only target those cancerous cells, leaving healthy cells alone.
Wow, that would be a game changer. Fewer side effects, more effective treatments. What other possibilities are there? I feel like we're just scratching the surface here.
Oh, we are. Think about autoimmune diseases, where the immune system mistakenly attacks the body's own tissues. If we understood the gene expression patterns in those immune cells, maybe we could reprogram them, stop them from attacking healthy tissue.
It's like hitting the reset button on the immune system, teaching it to recognize friend from foe again. What about inherited diseases? Could gene expression play a role there?
Absolutely. Often those diseases are caused by mutations in certain genes. By understanding how those mutations affect gene expression, we could maybe develop gene therapies to correct those errors, or even compensate for them by targeting other genes in the pathway.
So it's like we could actually edit the genetic code, fix those errors.
It's pretty amazing to think about the potential, right? And it's not just about treating diseases either. Understanding gene expression could completely change preventative medicine. Imagine if we could analyze someone's gene expression profile and identify potential health risks years in advance.
Whoa. It's like getting a glimpse into the future of your health.
Exactly. You could make lifestyle changes or receive early interventions based on your unique genetic predispositions, preventing diseases before they even start. That's the true promise of personalized medicine.
This is incredible, but it also makes me wonder about the bigger picture. We've been focusing on how genes are expressed, but what does this all mean for how we think about the role of genes themselves? Do our genes still determine who we are? Like, is it all pre-programmed?
That's a deep question. And it really challenges that old idea that our genes are our destiny. It's not just about the genes you inherit. It's about how those genes are expressed. And that can be influenced by a ton of things, like the environment you live in, the choices you make.
So it's not nature versus nurture. It's more like nature through nurture.
Exactly. Our genes provide the basic blueprint, but the environment plays a huge role in shaping how that blueprint is interpreted and expressed.
So does this mean our genes aren't as important as we once thought?
Not at all. Genes are still fundamentally important, but understanding gene expression gives us a more nuanced perspective. It's not just about whether you have a gene or not. It's about how much, when, and where that gene is expressed.
I see. So it's not enough to know what genes are in the library. It's about knowing which books are being read right now and how they're influencing the story.
Perfect analogy. Gene expression is like selectively reading those books, choosing which ones to open and which ones to leave on the shelf.
Yeah.
And that choice, that act of gene expression, can be influenced by all sorts of factors.
This is so empowering. It means we're not just puppets of our genes. We actually have some control over how those genes are expressed.
Absolutely. And that control can come from all sorts of things. Diet, exercise, stress management, even our social interactions. These can all impact gene expression.
So it's like our genes are not our destiny, but they do provide the framework. And by understanding how gene expression works, we can make informed choices to positively influence our health and well-being.
You got it. Knowledge is power, and understanding gene expression gives us the power to make choices that support our health goals.
This has been an incredible journey so far. We've learned that gene expression is so much more complex and dynamic than we ever imagined. There are all these layers of control, feedback loops, and even a system for buffering against variations. And as we learn more about gene expression, the potential for personalized medicine just keeps growing.
It's a really exciting time to be in this field. It has the potential to completely transform our lives and how we think about health and disease. And it all starts with understanding those fundamental principles of gene expression.
So, to our listeners, we encourage you to keep exploring this fascinating world. There's so much more to discover. And before we wrap things up, I have one final thought to leave you with. Something to ponder as you continue your journey of knowledge seeking. We've spent this whole deep dive exploring, how genes express themselves, this whole intricate dance of DNA to RNA to protein. But it makes me wonder, what does this all mean for the future of science itself? Research. Where do we go from here?
Oh, that's where it gets really exciting. This new understanding of gene expression, it's pushing scientists to think beyond just cataloging genes. It's like we've been focused on the words in a dictionary, but now we're starting to understand the grammar, the syntax, the meaning behind those words.
So instead of just knowing what genes exist, we're figuring out how they actually work together, how they interact, how they respond to their environment.
Exactly. And this is leading to all sorts of new avenues of research. For instance, scientists are now building these really complex computer models to simulate gene expression, like virtual cells.
Oh, wow. So they can test out different scenarios in these virtual cells.
Yeah, they can play around with gene expression, tweak things, and see what happens, all without needing actual physical cells.
That's incredible. So what kind of applications are we talking about here?
Well, imagine being able to design, completely personalized therapies, right? Based on an individual's unique gene expression profile. We could potentially predict how someone will respond to a particular drug or treatment, based on their genes.
That's amazing. No more trial and error, just super targeted, effective treatments from the get-go.
Exactly. And it goes beyond treating diseases, too. Imagine engineering crops that are more resistant to drought or pests, for example, or crops that produce way more food, all by manipulating their gene expression.
It's like we're unlocking the secrets of life itself. The possibilities seem endless. But with all this potential, I can't help but think about the ethical side of things. We're talking about manipulating genes, changing the very essence of living things.
You bring up a really important point. As with any powerful technology, there are ethical implications we need to think about very carefully. It's so important that we proceed thoughtfully and responsibly. We need to have those open conversations about the potential benefits and the risks of manipulating gene expression.
It's that old saying, right? With great power comes great responsibility.
Exactly. We all have a responsibility to make sure that these incredible advancements are used for good, to benefit humanity and the planet, not for harm or exploitation.
Well said. This has been a truly mind-blowing deep dive into the world of gene expression. We started with the basic definition, and then, wow, we went deep into this intricate world of regulation, buffering, and the incredible possibilities for personalized medicine and beyond. It's amazing to think that this whole universe of knowledge is hidden within our very cells, and it's constantly shaping who we are and who we can be.
It's a testament to the power of scientific curiosity, to that human drive to understand the world around us. And as we keep exploring gene expression, who knows what amazing discoveries we'll make? Discoveries that could change the future of science, medicine, and maybe even humanity itself.
That's the beauty of it all, right? The constant exploration, the never-ending quest for knowledge. So to all our listeners out there, keep those curious minds engaged, keep asking questions, keep learning. And we'll be back soon with another deep dive to uncover even more fascinating insights from the world of knowledge. Until then, happy exploring.